Did I give the name who discovered ribosome? Halliday. Halliday was the scientist name who discovered ribosomes. Okay. And of course, Swedberg was the scientist name who gave the molecular weight calculation for ribosomes. That's all. Then we'll get into the functional aspect of ribosome. You take the function of ribosome, basic function is nothing but protein synthesis. Double-stranded DNA molecule. Okay. Then while discussing about the DNA molecule, I explained you'll be having three components in DNA molecule. A nitrogen base. Okay. Then you'll be having deoxyribose sugar. Then a phosphate. So that's all makes the DNA. DNA. Deoxyribose nucleic acid. Okay. Deoxyribose nucleic acid. These are the three components. Now ribose, simple sugar structure. Phosphate, you know the phosphate molecule. But what about the nitrogen base? We will be having 4 nitrogen bases in DNA. They are A, T, G, C. These are the different nitrogen bases we'll refer to. I'm giving the list of these names over here on the other side of the slide. A stands for adenine. These are the special nitrogen bases we'll find only in nucleic acids. Okay, adenine. Then G stands for guanine. T stands for thymine. Don't get confused with this thymine with the B complex thymine. Okay. C stands for cytosine. Adenine, guanine, thymine, cytosine. Okay. Now these are the different bases you will find on DNA. They'll be arranged in different forms. Sometimes A will be there, sometimes G will be there, sometimes T will be there, sometimes C will be there. How that gets differentiated, like which molecule is there, which molecule is there on the DNA. Suppose if you take this as a DNA molecule, you will be having nitrogen arranged like this. Okay. On the strands, on the DNA strands, you will be having nitrogen bases arranged like this. The nitrogen bases may be A, G, T, A, C, G, T. It varies, it depends. But only these 4 only present throughout the structure. So assume that this is a DNA. Okay. Now, from DNA, you will get synthesis of RNA. RNA is different. Okay. When you take RNA, RNA stands for ribonucleic acids. See, there's only one difference. DNA deoxyribose, RNA ribose, ribonucleic acids. Now, RNA may be different types. It may be mRNA. It may be tRNA. M stands for messenger, T stands for transfer. Messenger RNA or transfer RNA. Based on the function, RNA may be mRNA or tRNA. Now here from DNA, we will get mRNA molecule. This is called transcription process. This is the term. This is the biology where a DNA molecule will synthesize RNA molecule, specifically mRNA. How it gets synthesized? See, here in RNA also you will have nitrogen bases and ribose sugar and phosphate like DNA is having. Now assume that here you are having A, U, G. Okay, here A transfer uracil, new term. You'll find only in RNA. I'm giving color discrimination so that you can understand. I gave the nitrogen bases in green color. Adenine, guanine, thymine, cytosine. These are the nitrogen bases in DNA. When you come to RNA, you will have adenine, guanine, thymine, but instead of cytosine, you will have uracil. Okay, this is another nitrogen base. So the nitrogen bases here produced are AUG, assume. How this AUG nitrogen bases are produced? Based on the nitrogen bases here on DNA. Okay, assume that here on DNA you are having TCA. So for T, there is synthesis of A. For C, there is synthesis of U. For A, there is synthesis of G. Like this it will happen. For one nitrogen base, there will be a component nitrogen base on RNA. So this we called as transcription. So the basic point over here is based on the type of arrangement on DNA, you will have type of arrangement in RNA formation. And later this RNA molecule will synthesize protein. This process we call as translation. Here see, this is mRNA. Okay. On mRNA, you are having various colored blocks. Can you see those blocks? Different colored blocks are there. Okay. Now these colored blocks are nothing but indicating the nitrogen base.
Yes, ma'am.
What happened?
Ma'am, I just said yes, ma'am.
Okay, fine. Thank you. So here, nitrogen bases, different nitrogen bases. Now I told you will be having different nitrogen bases like adenine, guanine, thymine, cytosine. Okay. Now here see, this is the RNA molecule which is produced. This RNA molecule is produced from DNA. How? Based on the nitrogen bases present on DNA. The complementary nitrogen bases are produced in RNA. Okay. Now when you take the DNA structure, you will be having different nitrogen bases. Based on that nitrogen bases, coding happens. The best word you want to go is nothing but coding. Based on the nitrogen bases, new nitrogen bases are produced in RNA. So this is how RNA is produced. Now, this is the ribosome, the bigger subunit and the smaller subunit. See, the mRNA comes and attach it to the smaller subunit of ribosome. And mRNA is having some codes. Now, this is a tRNA, transfer RNA. Just told you. Okay. The RNA molecule which transfers the amino acid. This is the amino acid. Okay. The amino acid towards the ribosome so that the code on the RNA, the nitrogen base code on the RNA gets decoded as amino acid and protein will be produced. This is called translation. Nothing but protein synthesis. Simple word, see, translation. Translate what? Suppose if a language is getting translated, how it will be translated? Based on the code, isn't it? How the language gets translated, same way here the nitrogen bases gets translated into protein. That's called protein synthesis. Okay? And I introduced another word, transcription. Transcription means what? That's nothing but decoding. So here this process is called transcription, where the DNA molecule is transcripted to RNA molecule. It's all happening. Only understand the process, DNA to RNA to protein. Okay. This is the central dogma of molecular biology. What are molecular biology? We do research for 3 years, 4 years. I gave it an instance. DNA to RNA, RNA to protein. This won't look like this much simple. If you want to understand this whole central dogma, you need to go for PhD. Okay, don't bother about the whole process. Understand the backbone. This is the backbone. DNA molecule, nitrogen bases gets coded into RNA molecule. RNA molecule, nitrogen bases gets decoded into protein. That's all. This all happens because of the ribosome, where the protein synthesis happens. Translation. Translation is nothing but translating. Translating of what? The codes which are there in mRNA. The nitrogen base codes. Those will be decoded into amino acids. All those amino acids will form protein. Nothing but translation. Translating the code on mRNA, into decoded form. The decoded form is nothing but protein. This process we call as translation. Clear? With amino acids, if you take this is an amino acid 1, this is amino acid 2, amino acid 3, amino acid 4, so forth. So like that, each and individual amino acids will be produced. See here, one amino acid, second amino acid. Like this, amino acids will be produced, and each amino acid are joined together with covalent bonds. I hope you know everything about covalent bonding.
Yes, ma'am.
So with the covalent sharing of electrons equally. So covalent bonding will be formed between different amino acids, and that forms the protein. Protein is nothing but a chain of amino acids. That's what we are saying, that DNA will be coded to RNA, and RNA gets decoded to protein. This the whole molecular biology over here. Now see, DNA will be having particular arrangement of nitrogen on the DNA structure, isn't it? Just like shown. Assume that this is the particular arrangement. This we call as code. Okay? This has to be coded as RNA. This is a normal process. In common, it will happen in general. But suppose if there is a sudden change, we call it sudden change. If there is sudden change in decoding this nitrogen base code of DNA, assume that this has to be coded as one protein. Assume that a protein A1. Okay, through RNA process and protein formation. But there might be some sudden change happening in this process where this code is wrongly interpreted. Using the word wrongly interpreted. What happens? Instead of production of A1 protein, some X1 protein will be produced. So this protein will not be produced because the code has been wrongly interpreted. The RNA structure would have changed. And instead of production of A1 protein, there might be production of X1 protein, which may become problematic to the organism. So if everything goes smoothly in a correct way, happy. But if there is change and that change is wrong change, then that is called a defect, isn't it? So this is normal process which has to happen, but because of sudden change, there may be a defect, which we call in biology as mutation. Mutation, by definition, goes like this. Mutation are the sudden changes in gene production or protein production. How the sudden change happens, that all will be discussed under mutation. That's a separate concept in molecular biology. Just understand how mutation is happening here. As you know the central dogma, here DNA has to be coded, RNA has to be formed, a protein has to be formed. But someday, some change happened. Because of that, wrong protein is produced, which will be a defect one. This you can observe in various disease conditions. Best example, cancer. See, in cancer, what happens? I gave just 2 words about cancer. There will be continuous division of cancer. There will be so many cells production cancer condition. So how that happens? Because of mutation. So normally, if 100 cells has to be produced in a particular cell tissue, there will be production of more than billion cells. Because of mutation, because of sudden change, number of cells increases. That's called cancer.
Ma'am,
Yes.
ma'am, but DNA are used in the, ma'am, it transfer the changes of the parents to the
That is
offspring.
completely related to nucleus. That is completely reproductive process related to nucleus.
Ma'am,
Okay.
it also uses the DNA, but
We have the DNA that is related to cytoplasmic inheritance. We discussed the maize and the yeast cell on the other session.
okay, ma'am.
Without reproduction also some characters gets transferred. That's because of cytoplasmic DNA, which is there in mitochondria and even chloroplast. Clear?
Yes, ma'am.
Reproductive method of inheritance happens only with nucleus. Non-reproductive method of inheritance may happen with cytoplasmic DNA. Clear?